So I'm going to try to ramp up from the sort of recordings that I do, but I like to consider, if you take the one of the turning points of the 20th century, the discovery of the double helix, and just stand back from that kind of discovery and imagine what a difference that made to our understanding of life itself. I think that my take on it would be that it wasn't just that was an important thing from a low-level point of view, but it gave high-level insights into the way genetics work, and I think we're still having high-level insights from thinking about the not just the structure of DNA, but everything, the epigenome, and all of the bits and pieces, the molecules, the procedures that happen at that level, don't just tell us how to do molecular biology, but also give us, I think, insights into at higher levels. So the argument I'm making is that I think you ought to be able to do the same thing with neurons in the brain, and that and in a way I think we've been duped by a reductionist approach to the brain, basically treating neurons as simple as we can and seeing what you can get out of it. And actually it's been remarkably successful. In a way I think it's a little bit illusory in that sense, because when you turn to the brain, it's clearly using elements that are much more sophisticated, and already that's been alluded to today. Iota Puerasi was, for instance, pointing out that it's now turning out that you can treat the dendritic tree as a deep neural network in itself, or at least it's got that kind of complexity. And so why does the brain take this strategy, if you like, to solving the cortex? So I want to take that seriously, and I've always wanted to take that seriously. I'm a kind of bottom-up scientist, and we started looking at neurons, and trying to work out why they are the way they are. But I think that you can see, if you extrapolate from the low-level kinds of explanations of what's going on, you ought to be able to, that ought to give you insights, just in the way that I think looking at the double helix does. So I'm hoping that out of this talk, I'm going to focus on what happens at the single neuron level when you go from being aware to not being aware and back again, and in particular what happens when you're anesthetized versus when you're conscious, and to look at this at the low level. Now, I know there are people here that have taken this from the top-down approach, and I hope that we can find a way to meet in the middle somehow. And so I'm hoping this talk, in some sense, brings these kinds of threads together. So one way of looking at what happens at the moment of consciousness is the so-called perceptual threshold. And this is a really old experiment in which you do some kind of stimulus that you control very precisely, and you ask a human subject, in this case, whether or not they feel the stimulus. And so it's been known for a long time that, first of all, when during a subliminal stimulus, a stimulus with not enough strength that you can feel it, there's still stuff going on in the brain. It's just that you're not conscious of it. And it does make sense, I think, to ask, so what happens in the brain when the person says they feel it? And to a first approximation, there are subsequent volleys that you can, or waves that you can record in the EEG that are, if you like, correlates for the moment of perception. And this can be done in animals as well, and one famous experiment was done in monkeys, and here you've got a little bit better access to the recordings from different neurons. They did this now 3 decades ago with an electrode array looking at the depths of the cortex, and basically they came to the conclusion that there are things going on in that second wave that correlates to consciousness. So in this second volley, that relates to information that's feeding back from higher cortical areas back to lower cortical areas, and they thought that this was inserting into the top layers of the cortex. So I'm going to try and convince you today that this correlates to a dendritic calcium spike, of all things. And that's what we've been focusing on for a long time. But just in advance of doing that, I should say a long-standing observation of the long-range connectivity in the cortex is that feed-forward information tends to hit the middle layers of the cortex, in particular layer 4, and the feedback information tends to avoid that and go to the outer regions of the cortex. And when you put the pyramidal neuron in this context, you can see then that I think it gives a clue as to why the cortex is ubiquitously pyramidal neurons, whichever area you look at. And the first thing I think we should expect is that the information that is going forward is going to mostly affect the bottom of these neurons, and the information that's going back will mostly affect the tufts. And this is a combination of the morphology of the pyramidal neurons, their orientation in the cortex, the fact that they're ubiquitous across the whole cortex, and this long-range architecture that the more we learn from institutions like the Allen Brain now and transgenic animals, it turns out that Fellerman and Van Essen, who first saw this, or first analyzed this in lots of data, in a sense, the first attempt to do what the human brain is now trying to do with mass data. I think that's now showing, proving to be completely correct, that to a first approximation, feedback is targeting at least the distal dendrites of pyramidal neurons. So one way of construing this is that the pyramidal neurons are basically shaped the way they are so that they can make exactly this comparison between feedback and feed-forward. And that's something we've been looking at for some time. And we've known also for a long time that whereas if you look at if you would stimulate the bottom of a neuron, of a pyramidal neuron, you'll typically get a firing train like this. And if you record in the dendrite, which is this red trace here, you'll see some reflection of that in the dendrite. But on the other hand, and I don't know how many people are aware of this. I showed this to some PhD students just a few weeks ago. They were admittedly all molecular biologists, but when I showed them this trace and asked them who actually knew about this phenomenon, nobody put up their hand. So I really don't know what to expect. I happen to know a lot of people in the audience who do know this particular fact, but I'd be interested to know how many people have never seen this before or never heard of the fact that there's a different kind of action potential in pyramidal neurons, and it's astoundingly strong. So it can drive the neuron in a completely different way. You can see these are the so-called plateau potentials that people refer to in the dendrites. It completely changes the output of the neuron. And I think you can see immediately that it really should matter whether or not there's feed-forward information impinging on these neurons or feedback information impinging on these neurons. So this is a so-called calcium spike, and it's something that we've been looking for under various circumstances. Going back to this situation, so we could essentially we're predicting that in the context of a stimulus that goes from being unperceptible to perceptible, that you'll start to evoke calcium spikes in the dendrites. And so we basically that's set up like this. We can do this in mice, and we're going to go through stimulus intensities of, in this case, moving the whisker just so much that you can't feel it. By definition, the perceptual threshold is when the animal will react 50% of the time to that stimulus strength. And then, of course, you can go to stimulus strengths where they get it up to 90% or better of the time. So we're predicting that, or we predicted some time ago now that when the stimulus was subliminal, you would not see calcium spikes in the appropriate pyramidal neurons, and on the other hand, when on the other side of the perceptual threshold, you would see this. And of course, what we really want to do is look at the perceptual threshold. So essentially, we do a two-photon imaging. Good news is that we're looking for calcium spikes, which are readily detectable, and these are plateau potentials, so even more calcium than you'd normally expect with activity in a neuron. And we can choose the height at which you expect to see calcium spikes in these neurons. And we get lots and lots of data. These are all the apical dendrites going through the plane of information, plane of view. And so we get lots of data for any given recording. And of course, we can precisely tell from the behavior of the animal whether whether it felt the stimulus. And what we're interested in is exactly this stimulus intensity, where half of the time the animal perceives it and half of the time it doesn't. And the upshot was, it's now a fairly old experiment, that when the animal perceived the experiment, there's a population of pyramidal neurons which robustly have calcium spikes and a population that don't. Of course, that's just a correlation between the dendritic calcium and the what we think is the perception of the animal. Nevertheless, I think it would count as a neurocorrelative consciousness. What we really wanted to do was manipulate that and see if we could manipulate the perception of the animal. And just to, I just conceptualize, so these are actual experiments but drawn in a conceptual way. So in other words, we had different strategies for inactivating calcium spikes in dendrites. And if we did that, we could shift the psychometric curve, this is a measure of the perception of the animal, to the right. In other words, it got harder for the animal to perceive the stimulus. And I thought, even more amazing was if we put channel rhodopsin in these pyramidal neurons and then light on the distal tuft, then we could push perception in the other direction. And not only that, but at stimulus intensities of 0, in other words, where we essentially faked a stimulus, the animal hallucinated that stimulus in about 40% of the cases. This is done in mice. So which is perhaps what you'd expect if perception is to do with activating the apical dendrite. Okay, and lastly, in that scheme of experiments, another experiment we could try, and it's become now routine basically, because in mice you can pick your transgenic animal and now target the neurons of your choice. So it's now possible to get a transgenic mouse in which you can look at cortical or intratelencephalic pyramidal neurons, or so-called pyramidal tract neurons, either the subcortically projecting neurons, and you can now do what you like with them essentially. And in these experiments we expressed alternately in these different transgenic animals a so-called dread, which allowed us to, these are basically the chemogenetic or the chemical version of an optogenetic experiment, which allows you to put the ligand, the chemical that binds to the receptors that you're now expressing anywhere you like, and you can now regulate the effect of these neurons. You can say what happens if these neurons cannot influence the downstream target where their axons are going. So by doing that, we found that in particular the higher order thalamic region was what was really necessary for conscious perception in these animals. Although it was also true of the superior colliculus and the striatum. The strongest effect we saw in the higher order thalamic region, which is for somatosensory stimuli in a rodent called the posterior medial nucleus, or the POM. But this is thought to be generally true of higher order thalamic regions. Interestingly, it had no effect to suppress the output of the pyramidal tract neurons on the brainstem, and we also couldn't influence the behavior of the animal, which we take to be its perception, by influencing the cortical or cortical striatal neurons, the intratelencephalic neurons. So it seemed to be special to the so-called pyramidal tract pyramidal neurons that they were somehow really intrinsically related. Their calcium spikes were very much related to detecting the stimuli and presumably perceiving it. All right, so with that, armed with that kind of knowledge, we then could do experiments that we were really interested in doing, which was, and basically we're now decades behind the people who've been doing these kinds of experiments in humans with various different approaches, EEG and fMRI and so on. But we could finally, with a hypothesis in mind, tackle the problem of anesthesia, which is, I'm sure we'll hear more about during these 3 days. And effectively, this is the flip side of consciousness in some sense. I think if we could understand how anesthesia works, we should be armed with some important knowledge for understanding consciousness. I guess that's about as far as I want to predict what we can do with the knowledge from anesthesia. But it's quite remarkable, anesthesia in itself. And just as a quick look at what we do know, this is more a statement of what we believe about consciousness in the first place, is that it seems to be be related to a large interreaction of information flowing in all directions, and we should discuss, I think, over the next few days whether this relates to some so-called ignition level, which is what Changeux and Dehaene would call the ignition point in a global neuronal workspace idea. And there are other ideas that relate to that. For instance, integrated information theory is another kind of way of looking at connectivity. There are specific predictions that are different from these different ways of looking at what consciousness might mean, but it does involve a kind of global interconnectedness. And in particular, when you lose consciousness, what seems to go away is feedback information in this equation. And this is some data from George Mashour's laboratory, in which they use different anesthesias, and they use methods for looking at feed-forward activity versus feedback activity. And you can see the blue stripes are where the different anesthetics are taking effect, and you can see that feedback information is killed in this manipulation. So there's something about feedback that's really intrinsic to what an anesthetic does, and presumably what's important for consciousness. All right, so getting back to this idea, we could finally now do the experiment that we really want to do. We've got transgenic animals. You can put channel rhodopsin in whatever neurons we like. So we pick the PT neurons that I was just saying we think are important for consciousness, and we can express channel rhodopsin in those neurons and put blue light wherever we like. And so we can pretend now, we can do a kind of global question on what happens if you would activate, just like feedback does, the tufts of these pyramidal neurons. And in the first instance, we saw what we expected, which was if you activate these neurons in an awake animal, you get robust spiking and burst spiking at the cell bodies, which is what we would get in vitro, actually, if we stimulated the same compartment. I showed that at the beginning. But we were kind of astonished to find that in the anesthetized animal, it flatlines. So the thing that's so astonishing about this, which maybe isn't really immediately obvious, is that this is quite a biophysical experiment, because channel rhodopsin is in these neurons. So we're effectively depolarizing this tuft, even in this circumstance, come what may. So that it's not as though we're leaving the circuit to do something in this case. We've put anesthesia into the systemically in the animal, in this case isoflurane, but we're still depolarizing the tufts via channel rhodopsin. So this implies that despite the fact that this part of the neuron is depolarized, the information just can't get down the neuron and influence the output of the neuron. So when you think about it, by doing that, you're effectively cutting off the apical dendrite by this manipulation, it appears. And what we would call decoupling these neurons, so that they're coupled under the awake state and they're decoupled under the anesthetized state. And if you imagine this is happening now ubiquitously over the cortex, we're effectively cutting off all of the apical dendrites in one fell swoop. And this may well be the explanation for why anesthesia works. So we could look at that in the depths of the cortex. Now we could use an electrode and put it at different depths. In fact, we could also use the Michigan probe, which is the linear array that I showed before, that was done in monkeys and is sometimes done even in humans. The problem was we got a photoelectric effect from putting the light on the array. So we used a glass electrode at lots of depths. The upshot was that we could see, using this approach, exactly where the propagation failed. We could also control for the fact that our depolarization of the tufts was happening under every circumstance. But in the anesthetic, when the anesthetic was taking action, it just had no effect on the cell body. So there's some kind of coupling zone, we think, that is attacked, if you like, by the anesthetics. We actually tried different anesthetics. We tried urethane and as well as isoflurane and pentobarbital, and could see in each case that, no, sorry, not pentobarbital, ketamine, that we could see in each of these cases that the tuft was cut off from the cell body. So one way or another, although also these anesthetics have different targets, they're somehow all manifesting in terms of their ability to block the propagation along the dendrite. Okay, so we think under certain circumstances there's a generalized ineffectiveness of input from the top to the bottom, and it's something to do with inactivation through the coupling zone. So one thing that we could show is that if you block the thalamus, you also had this effect. Just so you can interpret the results that I'm going to show now, we also could do this in the extracellular recording. So as opposed to where this is a so-called juxtacellular recording where we can see from a single neuron what's going on. But it's actually better and easier, I think, to record from a population of pyramidal neurons. And here you can see this is a filtered, a frequency filtered extracellular response from a population of pyramidal neurons at this depth. And you can see that it's very easy to see the effect of the anesthetic there. We can, of course, also put this electrode in the top and see the extracellular field that you get from the dendrites in this circumstance. And then we could test this phenomenon. So just as a heads up, the higher order thalamus, which is sometimes called the matrix thalamus, projects in this way to the cortex. I was just contrasting here the way, what you normally get described in textbooks is the relay parts of the thalamus that target the middle of the cortex. In the barrel cortex, you can see that nicely hitting layer 4. But the higher order thalamus projects not just to layer 1, but also to layer 5A, that's just below the barrels in the cortex. And we also know that about half, 50% of the action of these projections is through not ionotropic glutamate receptors, but metabotropic glutamate receptors. And a lot of that work was done by Murray Sherman and others, which has no particular explanation as far as I know at this point. In any case, when we downregulated the higher order thalamic region in the rodent, with muscimol, which is a GABA A agonist, but basically a way to inhibit areas, we found that so we went from the control case, which is this red extracellular recording, to the purple case in the background there, which is showing that by downregulating the higher order thalamus, you are decoupling the neurons. So just to repeat what's going on, we're still doing the same experiment. There's channel rhodopsin in these neurons. We're putting light on the tuft, and we're asking, does that depolarization of the top of the neuron affect the bottom of the neuron? And this is the bottom of the neuron, and this is the top of the neuron. So the top of the neuron is not affected by blocking the higher order thalamus, but which is only, well, I shouldn't say that. Our channel rhodopsin depolarization of the top of the neuron is not affected by blocking the higher order thalamus, but our channel rhodopsin depolarization of the top of the neuron is having much less influence on the bottom of the neuron when we block the higher order thalamus. So something that the higher order thalamus is doing to maintain the coupling of the neurons, and because we know that there was a lot of metabotropic glutamate receptors involved here, we did the obvious thing, which was to block metabotropic glutamate receptors in an awake animal. Under those circumstances, so this is a control, this is not an anesthetic now, the animal is awake, and we put in the metabotropic glutamate receptor blockers in the middle of the cortex, and now it flatlines again. So something about the activation of metabotropic glutamate receptors, presumably by higher order thalamus, in the middle regions of the cortex, is maintaining the connection from the top to the bottom of the cortex. We found the same kind of effect with cholinergic modulation. It wasn't quite as strong as metabotropic glutamate, cholinergic tone seems to also be important for coupling. So this, we feel, may be a way to explain what the action of anesthetics do and why they have such a catastrophic effect on a very particular thing, which is consciousness, without, and this is one of the other conundrums of anesthesia, without affecting the general properties of the neuron. Essentially what we're saying is that effectively the thalamocortical loop is maintaining this connection, and there's a weak link across the two main compartments of the pyramidal neuron. This by itself normally sustains a loop of feed-forward and feedback connectivity around the brain, and by doing something that either takes away this maintenance of that coupling, or some other way to take away this coupling, you basically decouple feedback ubiquitously around the cortex, and at the same time you would leave the activity state of the bottom of the cortex in exactly the same way that it was before, and so feed-forward activity should not be affected by the anesthetics. So I think it's a handy way to resolve what had been some, I think, confusion about which should be the most important effects, whether they should be cortical or thalamocortical. Essentially, it's everything at the same time, and this would make sense of that in one fell blow. So just 5 minutes. Okay. Just trying to put that into context of consciousness. I would already mentioned, I think, the two leading theories of consciousness that are about information flow around the brain. And I think it's clear that with either of these approaches to consciousness, if you blocked this axis across the pyramidal neurons, you would have a catastrophic effect on consciousness. And we're calling this dendritic integration theory just to signify that you can't understand, I think, how this would work if you didn't take at least this simplistic operational view of pyramidal neurons, that the apical dendrite is important. So we've actually been talking about a theory along these lines for a long time before we thought to try the experiments with anesthesia. I won't go into it now for lack of time, but basically we think that in the end these neurons are detecting, they're coincidence detectors or associational units that can detect where there's an overlap of feed-forward and feedback, and you basically get an ensemble of neurons that tell you that there's a correspondence between feed-forward and feedback at any point. And in the end, taking that from the point of view of a column, what that essentially means is that what's going into the top of the neuron should be somehow contextual information. This is internally generated information. And what would be going to the bottom of the neuron would be to do with the local processing of that, the feed-forward information that's needed for that particular area of the column. So I don't have a lot of time to go through this, so I'll skip over this. But the general claim would be that what the information coming into the top of the neuron would be the neuron that the brain is predicting that this column needs, if you like, everything that's not that feature but is related to that feature. And it's, in some sense, it would approximate to your expectation or prediction of that. I'll just skip over the memory experiments that I might have showed you if I had enough time. I just put this in context then. So what we're basically saying is that if you, in terms of how the information would be, how you should consider information around the cortex under this kind of a perspective of how it works. On the one hand, I think you'd expect that the feature-specific information would be learnt this would be sort of learnt statistically. This would be essentially during some sort of window of time when an animal or a person is experiencing the world. You would gather statistics, and that should wire up your cortex in the sense that you'd get all the information that relates to the particular features that you should be sensitive to. But then after some time, well, first of all, that would ground you in the world, and so all of your columns would actually be attributable to things that you've experienced many times. But after that period, what you really want to have is access to the other compartment, which is basically your predictions about the world, and this should be much more labile. So if that's the way of looking at it, essentially you go to a point where this becomes very stable and this becomes very labile, and you could start to do very quick kind of associations between the different columns. And we would be predicting that what's coming out of the cortical column is basically something like detecting the probability that any given feature is in your perceptual space given the context. That should be the stable part, and this should be the labile part. And now we're adding to that there's a way to couple the top and the bottom of the neuron when you're conscious, and essentially this would mean that semantic memory you should expect to find in the distal tufts of pyramidal neurons across the brain, and that this should be your conscious memory, if you like. This is the one that you would have access to through your predictions, and you would be made, you'll be able to access it if the thalamus could direct the activation of metabotropic glutamate receptors to the middle. So, I'll just finish with this illustration then. So, if that's the way we can think of a cortical column and the way that awareness is working, essentially, that would mean that your perception would relate to your knowledge in some way. That basically the predictions you can make here would affect what you would guess is, for instance, this animal in this picture. If you would look at it closely and you have some better information, maybe now you could decide, okay, maybe they weren't stripes on a tiger, maybe this was just shadows made by the gait on a dog. Now you see something completely different because now you're using your knowledge, and it's basically saying, yes, what we're saying is it's totally from the architecture of the cortex you can interpret why it is that you perceive this differently, and when you have now new knowledge, if you like, or when something occurs to you, it'll actually change your perception. All right, I just have to thank all the people involved in this and you for your attention.